Have you ever peered into the intricate, often unseen world that makes scientific breakthroughs possible? Or maybe wondered what underpins every critical medical diagnosis? It's way more than just those eureka moments, isn't it? It's the meticulous foundational work in the lab that really makes it all happen. So today we're taking a deep dive into that very realm. The fundamental techniques that are, well, the bedrock of modern biochemistry and laboratory science. Our mission. We've got a stack of sources, everything from a leading university's bioanalytical course materials right through to insights from a lab equipment provider. And we're going to distill the most important nuggets of knowledge, plus some surprising facts, basically offering you a shortcut to being well-informed about this whole essential domain.
That's exactly right. And this deep dive, it isn't just about listing techniques. We're really aiming to give you a comprehensive understanding, what ensures lab quality, the essential prep techniques, and the powerful tools used to analyze biological samples. And it's all grounded in these detailed guides. We looked at lab quality control, bioanalytical methods, the real nitty-gritty.
Okay, let's unpack this then. We hear about scientific results all the time, but what's the real secret? How do we ensure those results are consistently trustworthy?
Well, it all begins with something called laboratory quality control. LQC.
LQC.
Precisely. It's a robust system, really designed to eliminate the risk of, non-conforming outcomes, getting things wrong. Its core purpose is safeguarding the accuracy, the reliability, and importantly, the timeliness of every lab result. And it does that by detecting and rectifying errors early on.
Okay, so finding problems before they snowball.
Exactly. And here's a crucial detail, something that's often overlooked. Your quality control materials, the known benchmarks, they absolutely must be treated identically to the actual samples throughout the entire process.
So no shortcuts with the control.
None.
Yeah.
It's fundamental. Otherwise, how do you know your control is truly reflective of what's happening with your samples?
That makes sense. And it highlights that errors aren't just random, are they? They tend to happen at specific points. Our sources break these down into, 3 stages?
That's right. 3 distinct stages. First up is the pre-analytical stage. This is where errors happen before any testing even begins.
Okay, like what?
Think about simple things like a sample mix-up or mislabeling, or even improper storage or transport. These seemingly minor slip-ups can completely invalidate a result down the line.
Wow, so how do labs fight that?
It really comes down to strict adherence to standard operating procedures, SOPs. Meticulous labeling is key. Specific codes, dates, sources, what tests are needed, proper transport containers, the right temperatures, that's vital. And for potentially infectious or toxic samples, you've got stringent rules like the IATA triple packaging regulations, clear warning labels, everything.
Double packaging, okay?
Yeah. And another smart practice, maybe a bit underestimated, is creating subsamples. Aliquots, we call them.
Like backups.
Exactly. A crucial backup if something goes wrong later, saves immense time, potentially resources too.
Right, so that's pre-analytical. What's next?
Then you move into the analytical stage. This is where errors happen during the actual testing or the experiment itself.
So in the thick of it.
Right. Could be using the wrong reagents, maybe equipment that's faulty or hasn't been calibrated properly, incorrect proportions in a mixture.
Or just not following the procedure.
Or simply not following the SOPs during the test itself. Yeah.
Okay. These stages really hammer home the need for a solid quality management system, don't they?
Absolutely essential. And our sources really dig into the key areas that make the system work. First, and maybe most importantly, personnel.
The people doing the work.
The most vital resource. This means regular, targeted training, keeping people motivated, performance appraisals, running proficiency tests. Regular meetings are also crucial, for sharing information, addressing concerns proactively.
Makes sense. What else?
Then there's equipment. It's the backbone of the lab, right? So you need documented inventory, detailed maintenance records, thorough training for any new instruments, and manuals need to be easily accessible, not locked away in some cabinet.
Right, easy access.
Then purchasing an inventory. Ideally, this should be centralized for things like reagents, media, supplies. You need written procedures for selecting the right stuff, ensuring quality and consistency.
Okay, so controlling what comes in.
Exactly.
Good.
And process control manages all the activities from pre-analytical right through to post-analytical. Enforcing best practices for everyone who handles the sample, even if they're outside the lab, like during collection or transport.
So the whole chain.
The whole chain needs solid documentation, a dedicated coordination team. And it's not static. Process improvement is vital. It's an ongoing cycle. Identify errors, implement corrective actions, update your SOPs, update the quality management system, and communicate those changes effectively.
It sounds like a constant learning process.
It has to be. And for labs providing services, customer service is key. Open communication with clients, feedback channels, interviews, questionnaires, handling complaints properly, and then analyzing that feedback for improvement. It's a loop.
Right. And the last piece.
Facilities and safety. This is absolutely non-negotiable. Labs have potentially harmful chemicals, maybe infectious materials, so stringent safety protocols are essential, protecting staff, the environment, the community.
Yeah, safety first, always.
It means comprehensive training, strict SOPs, clear emergency response plans, proper waste management, knowing where everything is, eye-wash stations, fire extinguishers, first aid, and how to use it. Regular drills help too.
So all this internal quality stuff,
Yep.
how does it translate to credibility on a bigger stage, like internationally?
Great question. That's where standards come in. We need to distinguish between a few terms. There's certification, which is basically written assurance that a product or service meets specific requirements.
Okay, like a stamp of approval for a specific thing.
Sort of. Then there's accreditation. This is a formal acknowledgement that an institution is competent to provide a service or produce something reliable. It's about the system.
Competence. Got it. And the third one.
Licensure. That's often simpler. It's government recognition allowing an institution to legally operate, permission to open the doors, basically.
Okay. So who sets these standards?
Key bodies include the World Health Organization, the WHO. They develop standards for disease-specific labs, like they even run proficiency testing programs for critical areas like polio labs.
Wow. Okay.
And then there's the big one, the International Organization for Standardization, ISO. They're the world's largest developer of standards. You might have heard of ISO 9001, that's for general quality management systems. But there are very specific ones too, like ISO IEC 17025, which is specifically for testing and calibration labs.
Right, 17025. I've seen that one mentioned.
Yeah.
What are the actual benefits of getting that kind of ISO certification or accreditation? Is it just bragging rights?
Oh, far from it. The benefits are huge. It genuinely expands market share because it boosts customer confidence significantly. It forces continuous internal quality improvement through regular audits and assessments. It demonstrably improves staff competencies. There's training, evaluations.
So better skills overall.
Definitely. And for many labs, it's a fundamental requirement to get certain types of funding. Crucially, it enhances the credibility of your results massively. And in some countries, you actually need it for government registration to even operate.
Okay, so it's really foundational for trust. Now, shifting gears a bit from these big quality systems, let's get down to the basics, the actual stuff in the test tubes. Solutions. We hear aqueous solutions all the time in biochemistry. What is it about plain old water that makes it so indispensable?
It's fascinating, isn't it? An aqueous solution is simply a homogeneous mixture where water's the solvent. But water is just unique. It's over 70% of living organisms. And its properties that bend V-shape, its polarity, its ability to form extensive hydrogen bonds.
Hydrogen bonds, right? Those are key.
Absolutely. It averages about 3.4 hydrogen bonds at room temp, compared to 4 in solid ice. This network lets it ionize easily and dissolve an incredible range of polar molecules. But beyond just dissolving things, water plays an integral role in the actual structure, stability, and function of biomolecules themselves, like how proteins fold, how enzymes catalyze reactions. It's actively involved.
Wow. It's not just a passive background.
Not at all. It's a key player.
So given that we're making solutions in the lab, we need efficiency, precision. Why do we have different types of solutions, like stock solutions?
Right. We often start with stock solutions. These are prepared at a much higher concentration than we actually need for the experiment.
Why bother with the extra step?
Several reasons. It saves space. You store one concentrated bottle instead of many dilute ones. It saves preparation time later. It can minimize experimental variations batch to batch. And it makes it much easier to prepare solutions with really tiny final concentrations, like parts per million. Diluting is easier than weighing out minuscule amounts.
Okay, so stock is the concentrated form, then we make.
Working solutions, those are what you actually use in your experiment, diluting down from the stock.
Got it. And what about standard solutions? Is that different?
Yes, there's a distinction. While a stock solution is just concentrated, a standard solution has a precisely known concentration of a specific substance, an analyte. It's specifically made for quantification, calibrating an instrument or a test. Think of a standard D-glucose solution for calibrating a blood sugar test versus just a bottle of concentrated hydrochloric acid stock.
Okay. Precision for measurement is the key difference.
Exactly.
And how do we talk about these concentrations? I know there are lots of units.
There are quite a few, each useful in different contexts. You've got percentage, which can be weight by volume, WV, or volume by volume. Pretty straightforward. And then molarity, M, moles of solute per liter of solution. Very common, but it's temperature dependent because liquid volume changes with temperature.
Right, so you need to know the temperature. That's where M1, V1, M2, V2 comes in handy for dilutions, right?
Precisely. A very useful calculation. Then there's normality, which is gram equivalents per liter. Also temperature dependent.
Okay. Is there one that isn't affected by temperature?
Yes, molality M. That's moles of solute per kilogram of solvent. Because mass doesn't change with temperature, molality is temperature independent. More precise for certain physical chemistry applications.
Molality. Okay, good to know.
Yeah.
What about really tiny amounts?
For varied dilute solutions, we often use parts per million, ppm. For aqueous solutions, it's often equivalent to milligrams per liter, mgl.
ppm, right. And the last one?
Mole fraction. This is just the ratio of the moles of one component, say the solute, to the total moles of all components in the solution, unitless.
Okay, lots of ways to measure concentration. Speaking of which, let's zoom in on pH. It's fundamental, but can be confusing. What's the simple take?
pH is simply the negative logarithm, base 10, of the hydrogen ion concentration, H+.
Okay, negative log. Why the scale 0 to 14?
That comes from the ion product of water, Kw. At 25 degrees C, Kw is H plus times OH, and it equals 1 times 10 to the minus 14m squared. That constant relationship across the balance of H plus and OH ions naturally leads to the 0-14 scale.
Ah, it's tied to water's own chemistry. And we measure it with.
For precise values, we use a pH meter. But for quick, approximate readings, simple indicator dyes or pH paper work fine.
And clinically, getting pH right is critical, isn't it?
Absolutely vital. Normal human blood pH is tightly regulated around 7.4. If it drops too low, maybe below 7.35, that's acidosis. Can happen in severe diabetes, for example.
And if it goes too high?
That's alkalosis, maybe above 7.45. Both acidosis and alkalosis can be life-threatening if they aren't corrected.
Wow. And it affects more than just blood, right?
Oh, yes. pH profoundly impacts the structure and therefore the function of macromolecules like enzymes and proteins. Change the pH, you can change their shape, maybe denature them, stop them working. It's crucial for maintaining normal physiological functions everywhere in the body.
So if pH is so critical and so sensitive, how do biological systems and our experiments stop it from swinging wildly all the time?
That's where buffers come in. Brilliant things. They're aqueous solutions specifically designed to resist changes in pH when you add an acid or a base.
How do they do that?
They're typically mixtures of a weak acid and its conjugate base, like acetic acid and sodium acetate, for example. This pair can sort of soak up excess H plus or OH ions, minimizing the pH shift. They work best within their specific buffering zone, which is usually about 1 pH unit above and below their pKa value.
And our bodies use these?
Constantly. We have powerful biological buffer systems, like the phosphate buffer system and the bicarbonate buffer system in our blood. Even proteins themselves can act as buffers, especially amino acid residues like histidine, which buffers well near neutral pH. Nucleotides, other metabolites too. It's a complex system.
And there's an equation for this, right? Henderson-Hasselbalch.
That's the one. It describes the relationship between pH, pKa, and the ratio of the conjugate base to the weak acid. Very useful for buffer calculations.
Okay. Buffers are key for stability.
Yeah.
Now, given all these chemicals procedures, safety has to be paramount.
Cannot be overstated. Every single person in a lab, worker, student, visitor, and the institution itself must adhere to strict safety rules. The potential hazards are real, from chemicals to infectious materials.
It's about protecting everyone.
Yourself, your colleagues, the environment, the community, absolutely.
So what are some non-negotiable safety practices?
Okay, top of the list. Always attend safety training. Follow instructions and SOPs to the letter. Read labels and procedures carefully before you start anything. Report damaged equipment immediately. Don't try to use it. Never leave experiments unattended. Know where all the emergency equipment is: eye wash, safety shower, first aid, fire extinguisher, and how to use it.
Right, know your exits too.
Exactly. Handle sharps needles, glass with extreme care. Use fume hoods for hazardous or volatile substances. Only do the experiments you're actually instructed to do. Don't improvise dangerously.
And simple things like hot stuff.
Always use appropriate holders for hot objects. Never grab them directly.
Common sense, but critical. What about personal protection?
Fundamental. Always wear your lab coat. Always wear disposable gloves when handling samples or chemicals. Always wear safety glasses or goggles.
No exceptions.
No exceptions.
Yeah.
And no eating, drinking, or mouth pipetting in the lab, ever. And never use lab glassware for food or drink. That's a huge contamination risk, both ways.
Yeah, definitely don't want to drink from a beaker.
Please don't tie back long hair. No open-toed footwear shoes must cover your feet completely. No food stored in lab refrigerators.
And don't work alone.
Never work alone, especially when doing potentially hazardous procedures. Wash hands thoroughly after removing gloves and before leaving the lab. Always cover any open wounds before starting work.
Okay. Any specific chemical handling tips?
A classic one, always worth repeating. When diluting concentrated acids, always add the acid slowly to the water, stirring constantly. Never, ever add water to the acid.
Why is that?
The mixing process, the hydration, is highly exothermic. It releases a lot of heat. Adding water to acid can cause it to boil and splash dangerously. Acid to water, A to W.
A to W. Got it. And finally, waste.
Proper waste disposal is key. Waste must be segregated: sharps, chemical, biological, regular trash, and clearly labeled according to institutional and regulatory policies. You can't just dump everything down the sink or in the bin.
Right. Okay, so we've built this foundation of trust. Through quality control, we understand our solutions. What happens when we need to actually get inside the cells or tissues? Our next step in this deep dive is exploring how scientists literally break open cells and then start separating out the interesting bits inside.
Exactly. The whole purpose of cell disruption and extraction is to get at those intracellular components, things like proteins, enzymes, hormones, DNA, RNA, whether they're naturally there or maybe recombinant proteins we've engineered the cell to make.
So how do you crack open a cell? What's interesting here is that the sources mention two main strategies: non-mechanical and mechanical. Let's start with non-mechanical.
Right. Non-mechanical methods often use chemical or biological means. Chemical methods, for instance, might involve organic solvents like alcohols or toluene that can dissolve cell membranes. Or detergents are very common. Things like Triton X100 are good for gently solubilizing membrane proteins, while a stronger one like SDS will basically blow the cell apart and denature proteins, which might be what you want sometimes.
Depends on the goal. What about biological tools?
That's enzymatic methods, using specific enzymes to break down specific structures, like lysozyme for certain bacterial cell walls, or cellulases for plant cell walls, chitinases for fungi. Highly specific, which can be a big advantage.
More targeted than just dissolving everything.
Exactly. And then there's the simplest, maybe, osmotic shock. Just changing the external environment drastically, causing the cell to swell up and burst due to water rushing in or out. Works well for some cells, like red blood cells.
Okay, those are the gentler, or at least non-physical methods. What about the mechanical ones?
Yeah.
Sounds more forceful.
They generally involve physical forces, yeah. We can group them into solid shear methods. One example is the bead mill.
Bead mill.
Yeah, imagine a rotating cylinder partially filled with tiny, hard beads, often zirconia. As it spins, the beads collide and create shear forces that just grind the cells open. Advantages are high efficiency, you can control the temperature pretty well, and handle quite a lot of cell mass.
Okay, what else in solid shear?
The French press. This forces cells under incredibly high pressure, like up to 1,500 bar through a tiny valve. The sudden drop in pressure as they exit makes them explode.
Intense. Okay, what about liquid shear?
Right, liquid shear methods. High-pressure homogenization is common. Cells are forced through a narrow channel at high pressure, maybe 1,200 psi. This creates shear and cavitation bubble formation and collapse, breaking them up into tiny particles. It's fast, easy to scale up, but a big downside is the heat generated. Not good for heat-sensitive molecules.
So you might cook your protein.
You might. Then there's ultrasonication. Using high-frequency sound waves, usually around 25 kilohertz. These waves create cavitation, tiny bubbles forming and imploding violently, and that energy disrupts the cells. It has lower operating costs, and you can minimize protein denaturation if you keep it cold, do it in short bursts. But continuous sonication generates a lot of heat and can even create damaging free radicals.
So pulses are better.
Often, yes. Another method is freeze-thaw. This relies on temperature cycles. You freeze the sample, often rapidly in dry ice, then thaw it. Repeat this several times. The ice crystals forming and melting disrupt cell structures, causing them to break. It's time-consuming, but can be quite gentle, often used for isolating recombinant proteins from bacteria.
Okay, and one more.
The Potter-Elvegem homogenizer. This is simpler, often used for soft tissues or animal cells. It's basically a glass tube and a tight-fitting Teflon pestle, often driven by a variable speed motor. It grinds the tissue and shears the cells. It's inexpensive, and you can easily keep the sample cold in an ice bath during homogenization.
So lots of ways to break cells open, depending on the cell type and what you want to preserve. Once you have this cell soup, this lysate, you usually have a complex mixture. How do you start pulling out specific molecules?
That's where extraction techniques come in. Liquid extraction, or solvent extraction, is a classic. It works based on the different solubilities or affinities molecules have for two immiscible liquids, like oil and water, essentially. You mix your sample with two solvents that don't mix. Your target molecule will prefer one solvent phase over the other. You let the phases separate, collect the phase containing your molecule, and maybe wash or concentrate it. Widely used in pharma, environmental testing, food industry.
So choosing the right solvents is key.
Absolutely. And related to that is solid liquid extraction. Here, you're pulling compounds out of a solid material using a liquid solvent.
Like making coffee.
Exactly like making coffee. Or extracting active compounds from medicinal plants or flavors from food items. You need to choose the right solvent. Often grind the solid material to increase surface area, then perform the extraction methods like oxalate extraction, simple soaking, maceration, or percolation exist. Then you filter, concentrate, and analyze.
Okay, extraction helps simplify the mix, but for really precise separation, especially of larger things like organelles or proteins, we need something more powerful, right?
Most definitely. And that often means turning to one of the most fundamental tools in the biochem lab, centrifugation.
The spinning machine again.
The spinning machine, its principle is brilliantly simple. It just uses centrifugal force to accelerate the natural process of sedimentation, things settling out based on density and size.
Okay, but there's some specific terminology here. RCF.
Right. RCF stands for relative centrifugal force. It's the force applied to the sample compared to Earth's gravity, expressed in units of g. There's a formula. RCF equals roughly 1.12 times 10 to the 5 times the speed in RPM squared times the radius of the rotor in centimeters.
Okay, let's try an example. Say 20,000 RPM in a rotor with a 3.5 centimeter radius.
Plug that in. You get about 15,652 grams. So you're subjecting your sample to over 15,000 times the force of gravity.
Wow, that'll make things settle fast. And RPM is just.
RPM is simply revolutions per minute, just how fast the rotor is spinning. But RCF is the more scientifically relevant measure of the force being applied.
Got it. And Svedberg units.
The Svedberg unit S is a measure of sedimentation velocity, how fast something moves in a centrifugal field. One Svedberg unit is tiny, 1 by 10, 13 seconds. But it allows us to compare how quickly different molecules or particles sediment. A higher S value means that sediments faster, like bacterial ribosomes are 70S, while our eukaryotic ribosomes are 80S. They separate based on that difference.
Okay, Svedberg for sedimentation speed. So how does the centrifuge actually work? The components.
Well, you've got the motor that spins the rotor, where you place your samples in centrifuge tubes. There's a control panel to set speed and time, a secure lid, and often a cooling system, especially for high-speed ones, to stop samples overheating.
And the process.
You load your balanced sample tubes into the rotor, close the lid securely, set your desired RCF or RPM in time, and start the run. As it spins, the centrifugal force pushes denser, heavier particles towards the bottom of the tube, forming a solid pellet. Lighter particles in the liquid stay above, forming the supernatant. When the run stops, you carefully remove the tubes and pipette off the supernatant, leaving the pellet behind. Or maybe you want the supernatant.
Like separating blood.
Perfect example. Spin down whole blood, and you get the clear yellowish plasma on top. Then a thin whitish layer called the buffy coat, that's white blood cells and platelets, and finally a dense red pellet of erythrocytes, the red blood cells, at the bottom.
Clear separation. Do the rotors themselves differ?
Oh yes, different rotors for different jobs. A fixed angle rotor holds the tubes at a constant angle, maybe 14 to 40 degrees. Good for quickly pelleting material, common in differential centrifugation.
Okay, angle tubes.
Then there's the swinging bucket rotor. The tubes hang vertically when stopped, but swing out to a horizontal position when spinning. This is ideal when you need a really flat, even pellet, or for techniques like rate zonal centrifugation, where you're separating things into bands within the tube.
Horizontal spin, and the last type.
A vertical rotor. The tubes stay vertical throughout the spin. This gives the shortest possible path length for particles to travel, so runs can be faster. often used for specific types like isopycnic centrifugation.
Okay, different rotors. And the centrifuges themselves vary hugely in power, right?
Absolutely. The everyday benchtop centrifuge, you see them in clinical labs, maybe spinning blood tubes or cell cultures, relatively low speed, maybe 3,000 to 7,000 grams.
The workhorse.
Exactly. Then for smaller volumes, like zero point 0.5 to 1.5 milliliter tubes, you have the microcentrifuge, often called a microfuge, much faster, up to maybe 15,000 grams. Great for pelleting DNA or concentrating proteins quickly.
Microfuge, got it. And then the big guns.
Then you have the ultracentrifuge. These are the heavy hitters, capable of incredible speeds up to maybe 150,000 RPM, generating forces over 1,000,000 times gravity, 1,000,000 grams.
1,000,000 jam. That's insane.
It is. They always have refrigeration and a vacuum system inside the chamber. The vacuum reduces air friction, which would otherwise heat the rotor and sample massively at those speeds.
Makes sense. Are there different kinds of ultras?
Primarily two types. The preparative ultracentrifuge is used for isolating and purifying large amounts of material, separating subcellular organelles like mitochondria or ribosomes, purifying viruses, proteins, DNA, RNA.
For bulk separation.
Right. And then the analytical ultracentrifuge. This is more specialized. It has built-in optical systems detectors that can actually monitor the particles during the centrifugation run. This lets you measure things precisely, like sedimentation coefficients, molecular weight, even the shape of molecules in solution. Theodor Svedberg won his Nobel Prize using an early analytical ultracentrifuge to study hemoglobin.
Wow, real-time analysis while spinning at crazy speeds.
Pretty amazing technology.
So, beyond just pelleting stuff, what are the more advanced separation strategies using these machines?
Well, a classic technique is differential centrifugation. You take your cell lysate, your broken up cells, and spin it at a relatively low speed. The heaviest stuff, like nuclei and intact cells, pellets out. You collect the supernatant, spin it again, but faster. Now, maybe mitochondria and lysosomes pellet, collect that supernatant, spin even faster. Maybe microsomes pellet, and so on.
So separating fractions by increasing speed.
Exactly, sequentially separating components based roughly on size and density. The main drawback is purity fractions often get cross-contaminated with components from the previous or next spin speed.
How do you know what you've got in each pellet?
Often by assaying for specific marker enzymes known to be enriched in particular organelles. Like succinate dehydrogenase is a marker for mitochondria, acid phosphatase for lysosomes, cytochrome P450 for microsomes.
Okay, markers help identify. But what if you need higher purity?
Then you move to density gradient centrifugation. This is the solution to that cross-contamination problem. It separates particles based on much finer differences in size, mass, and density using a gradient medium.
A gradient, like thicker at the bottom.
Exactly. You create a solution in the centrifuge tube that gradually increases in density from top to bottom. Common gradient materials are sucrose or glycerol solutions, or things like cesium chloride for DNA.
And how does that help separate?
Two main ways. First is rate zonal centrifugation. Here, you carefully layer your sample on top of a preformed density gradient, like 5 to 20% sucrose. When you spin it, particles move through the gradient based primarily on their size and shape their sedimentation coefficient S value. Faster sedimenting particles move further down. They separate into distinct bands or zones, good for separating organelles, ribosomes, viruses.
So separation by speed through the gradient. What's the other way?
Isopycnic centrifugation, also called equilibrium density gradient centrifugation. Here, separation is based purely on buoyant density. You often mix your sample with the gradient material, like cesium chloride, and spin it very hard for a long time. The gradient forms during the spin, and particles migrate until they reach a point in the gradient where their own density exactly matches the density of the gradient medium.
So they float at their own density level.
Precisely. They form sharp bands at their isopycnic point. The beauty is, the separation is independent of sedimentation rate or molecular mass. It's only about density.
That sounds incredibly precise.
It is. The classic example is isolating DNA. DNA has a buoyant density around 1.70 grams per cubic centimeter. In a cesium chloride gradient, that density corresponds to about a 6 molar CSCl solution. So the DNA forms a sharp band right there, separated from proteins, lower density, and RNA, higher density, about 2.000 GCMO3.
Wow, that's elegant. You mentioned cesium chloride, sucrose,
Yeah.
any others?
Yeah, common gradient media include sucrose and glycerol for organelles and proteins, cesium chloride mainly for nucleic acids, and then proprietary media like ficol and percol, which are often used for separating cells because they are less osmotic.
Okay. Lots of options. Before we leave centrifuges, any final safety thoughts? They sound powerful.
Absolutely critical. Always, always balance the tubes in the rotor precisely. An imbalance, especially at high speeds, can destroy the machine. It's incredibly dangerous. Never open the lid while the rotor is moving. Never try to touch a moving rotor. If you hear strange noises or see excessive vibration, stop the run immediately. Keep the centrifuge, especially the rotor and chamber, clean and dry to prevent corrosion. Maintain a logbook. Rotors have a limited lifespan based on run hours and speed. And personal protection, lab coat, safety goggles, always.
Okay. Balance tubes, keep the lid closed, listen for problems.
Got it.
So we've prepared samples, maybe broken cells, separated components. But what if we want to actually see or quantify specific molecules based on their inherent properties, not just size or density?
Right. Now we move into the realm of light. Our final stop on this deep dive is spectrophotometry, using light to reveal the secrets hidden within a sample.
Spectroscopy, spectrophotometry. What's the difference?
Spectroscopy is the broad study of how matter interacts with electromagnetic radiation light, energy. It could be absorption, emission, scattering. Spectrophotometry is a specific analytical technique derived from spectroscopy, mainly focused on measuring how much light a substance absorbs at different wavelengths. It's a powerful tool for investigating molecular structure and quantifying biomolecules.
Okay. And just quickly, remind us about electromagnetic radiation.
Sure, it's a whole spectrum of waves, from high-energy X-rays and UV light through the visible light we see, down to lower-energy infrared and radio waves. They all have properties like wavelength, frequency, amplitude. And crucially, the energy of a photon is directly related to its frequency and inversely related to its wavelength.
Right. EO-SHU. So how does this light interact with molecules in our sample?
Two main ways are relevant here. One is light scattering, like Rayleigh scattering. Photons basically bounce off molecules, changing direction, but not energy or frequency. This can be useful, like an X-ray diffraction, to figure out structures, but it's not absorption.
Okay, bouncing off. What's the other way?
Absorption. This is where the energy of a photon is actually transferred to the molecule, exciting it to a higher energy state. Critically, molecules only absorb light of specific wavelengths, specific energies that match the energy difference between their electronic states. This creates a unique pattern of absorption across different wavelengths, an absorption spectrum.
Like a fingerprint.
Exactly like a molecular fingerprint. It's characteristic of the molecule's structure, especially its functional groups, and we can use it for identification. And for this, we need to differentiate between polychromatic light with many wavelengths, like sunlight, and monochromatic light filtered down to a single wavelength, which is what we use for precise measurements.
Okay, absorption spectra for identification.
Yeah.
How does this help us measure how much of something is there?
That brings us to the cornerstone of quantitative spectrophotometry, the Beer-Lambert law.
Sounds important. What does it say?
In simple terms, it states that the amount of light absorbed by a solution is directly proportional to two things: the concentration of the absorbing substance and the path length, the distance the light travels through the sample.
More stuff or a longer path means more light absorbed. Makes sense.
Precisely. We define transmittance T as the fraction of light that passes through the sample, intensity out intensity in IIO, and absorbance A, which is more commonly used, is defined as the negative logarithm, base 10, of transmittance. A equals a log 10 T. Absorbance is the value that's directly proportional to concentration.
Okay, A is proportional to concentration and path length. What's the equation?
The fundamental equation is A is absorbance, unitless. C is the concentration, usually in molarity. L is the path length, usually 1 centimeter for standard cuvettes. And epsilon.
Epsilon, what's that?
A is the molar extinction coefficient, sometimes called molar absorptivity. It's a constant for a given substance at a specific wavelength and in a specific solvent. It represents how strongly that substance absorbs light at that particular wavelength. A high epsilon means it absorbs very strongly, even at low concentrations.
And how is that useful?
Hugely useful. If you know the extinction coefficient A for your molecule at a certain wavelength and you measure the absorbance A of your solution in a 1 centimeter cuvette, so L1, you can directly calculate the concentration, CaO day. It's a direct way to quantify your substance.
Wow, okay, so knowing epsilon is key. Are there known values for common molecules?
Oh, yes. For example, proteins are often measured at 280 nanometers in the UV range because the aromatic amino acids tryptophan and tyrosine absorb strongly there. We know their average contribution to epsilon. Or important coenzymes like NADH and FAD have strong, distinct absorption peaks around 340 millimeters, which lets us easily monitor enzyme reactions involving them.
Okay, so ALA lets us quantify things. What kind of instrument does this?
That would be a UV visible spectrophotometer, incredibly common in biological research and chemical analysis, used for everything from basic quantification to measuring enzyme kinetics, screening drug candidates.
And it works by measuring absorption by, what did you call them? Chromosomes?
Chromophores, close. Chromophores are the specific parts of a molecule that actually absorb the UV or visible light. Could be double bonds, aromatic rings like in tryptophan, peptide bonds in proteins, though they absorb further in the UV, prosthetic groups like the porphyrin ring in heme, or those coenzymes NADH and FAD.
Chromophores. Got it. What's inside the machine?
Key components include a light source, typically a deuterium or hydrogen lamp for the UV range, roughly 200 to 400 millimeters, and a tungsten lamp for the visible range, 400 to 700 millimeter. This initial light is polychromatic. It then passes through a monochromator, usually a prism or a diffraction grating, which selects out a very narrow band of wavelengths, giving you monochromatic light.
So you can choose exactly which wavelength hits the sample.
Exactly. The monochromatic light then passes through the sample, which is held in a cuvette. Cuvettes need to be transparent at the wavelengths you're using. Glass is fine for visible light, but you need quartz or fused silica for UV work, as glass absorbs UV light.
Quartz for UV. Okay.
Yeah.
And after the sample.
The light that wasn't absorbed hits a detector. Traditionally, this was often a photomultiplier tube, PMT, which is very sensitive, and converts the light signal into an electrical signal that gets measured. Newer instruments often use photodiode arrays, PDAs, which can measure absorbance across a whole range of wavelengths simultaneously, much faster.
Faster measurements. Are there different setups? Single beam, double beam?
Yes. A single beam spectrophotometer measures the light intensity passing through the sample. You first have to measure a blank, usually your solvent in the same cuvette, and subtract that reading from your sample reading. A double beam instrument is more sophisticated. It splits the light beam, sending one path through the sample and the other simultaneously through a reference, blank. It automatically compares the two and directly gives you the absorbance of the sample, correcting for the solvent absorbance in real time.
Double beam sounds more convenient.
Generally is, especially if the solvent itself absorbs significantly or if the lamp intensity fluctuates. Applications are broad identification via spectra, quantitative estimation using Beer-Lambert.
Any limitations?
Sure. Sensitivity can be affected by sample pH or temperature sometimes. Turbidity or cloudiness in the sample will scatter light, giving falsely high absorbance readings. And the Beer-Lambert law itself can deviate at very high concentrations where molecular interactions interfere.
Okay, good to keep in mind. So that's absorption. What about fluorescence? You mentioned emission earlier.
Right, fluorescence spectrophotometry, or fluorometry, works on a different principle. Instead of just measuring absorbed light, we measure emitted light.
How does that work?
A molecule absorbs a photon of light, getting excited to a higher energy state, just like in absorption. But then instead of just losing that energy as heat, it relaxes back down to its ground state by emitting a photon of light. Crucially, this emitted photon always has lower energy and therefore a longer wavelength than the absorbed photon. Some energy is always lost before emission. This difference in wavelength between the absorption peak and the emission peak is called the Stokes shift.
Longer wavelength emission. Stokes shift. Okay. Is it efficient?
Not always. The efficiency of fluorescence is measured by its quantum yield, basically, the ratio of photons emitted to photons absorbed. Some molecules are highly fluorescent, high Q, others barely fluoresce at all, low Q.
So how do you measure this fluorescence? The instrument.
A fluorometer, or spectropluorometer, has some similar components but a key difference in geometry. You still have a light source, often a powerful xenon arc lamp, to provide intense light across a range of wavelengths. Then, an excitation monochromator selects the specific wavelength you want to use to excite your molecule. This light hits the sample in a cuvette. Then, critically, the detector and an emission monochromator are usually placed at a 90-degree angle to the excitation light path.
90 degrees. Why?
To minimize interference from the bright excitation light just passing straight through. By detecting at 90 degrees, you primarily collect the light that's been absorbed and then emitted by the sample in all directions, while most of the excitation beam goes straight ahead and misses the detector. The emission monochromator then selects the specific emission wavelength you want to measure.
Clever setup. What kinds of molecules fluoresce?
We talk about two types of fluors, or fluorophores. First are intrinsic fluors molecules that are naturally fluorescent.
Like what?
In biology, the main ones are the aromatic amino acids, especially tryptophan, but also tyrosine and phenylalanine to lesser extents. Also, some nucleotides and coenzymes like NAD and FAD have intrinsic fluorescence. We can use changes in their fluorescence to study protein folding or unfolding, probe enzyme active sites, follow kinetics, all without adding any external labels.
Using the molecule's own glow. Cool. What's the other type?
Extrinsic fluors. These are fluorescent dyes or labels that we deliberately add to a system. Things like dansyl chloride, fluorescein, rhodamine, ethidium bromide, which binds DNA, acridine orange, tons of them. These are often much brighter than intrinsic fluors and can be designed to bind specifically to certain molecules or cellular compartments. We use them for highly sensitive quantitative analysis, qualitative imaging, studying interactions like how a drug binds to DNA or a protein.
So adding a fluorescent tag, what are the broader uses?
Oh, huge range. Fluorescent microscopy is built on this, allowing us to visualize specific structures or molecules within cells with incredible contrast. Fluorescent antibody detection is used in diagnostics, like finding tumor markers or detecting viruses inside cells. Very sensitive quantitative DNA measurement often uses fluorescent dyes that bind to DNA, detecting and quantifying peptides and proteins. The list goes on. It's generally much more sensitive than absorbance spectrophotometry.
More sensitive. Any downsides?
The main one is that fluorescence is very sensitive to the local environment. Changes in temperature, pH, solvent polarity can significantly affect the fluorescence signal. And there's a phenomenon called quenching, where other molecules in the solution can interact with the fluorophore and decrease its fluorescence intensity, sometimes unexpectedly. So you need very careful experimental control.
Okay. Super sensitive, but needs careful handling. Wow. We've really covered a lot of ground today, haven't we? From the absolute necessity of meticulous lab quality control and getting solutions right, to the sometimes brute force methods of breaking cells open, the elegance of separating things with centrifuges, and finally using light itself, absorption, and fluorescence to see and measure molecules, you really do have a shortcut now to being genuinely well-informed about these foundational techniques that, well, they underpin so much of modern biochemistry.
Indeed. We've seen how it all comes together. Precision, safety, understanding the subtle properties of molecules, how they interact with forces like centrifugation or with light, from making sure that initial sample is handled perfectly, right through to isolating that one specific protein or determining a compound's concentration down to tiny amounts. These techniques really are the silent bedrock of biological discovery. They turn raw samples into actual knowledge.
So what does this all mean for you listening in? Maybe consider this. Now that you have a clearer picture of this meticulous journey, a sample takes, how it's prepared, purified, analyzed at such a fundamental level, what new questions does this raise? Questions about reliability, about innovation, maybe even about the future possibilities that are built upon this foundation in today's and tomorrow's biomedical research. Something to think about.